Hello, everyone, and welcome to today's webinar, The Good, the Bad, and the Ugly of 45-Color Panel Design, Building to Higher Dimensional Flow Cytometry While Maintaining Biological Resolution. I am Anson Blanks of Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Seddon Thomas, R&D Staff Scientist at Thermo Fisher Scientific.
All right. Thanks, Anson. Today, as Anson mentioned, I'm going to be talking about the good, the bad, and the ugly of 45-color panel design. And a lot of this is really thinking about, as you build to more and more colors, how do you maintain biological resolution? And so, when I initially started thinking about really how dyes affect your panel design, I had to step back and realize how much we're actually accounting for bad dye performance. So conventional dyes really only perform in two ways, and both can lead to spread, which is really loss of data resolution. And so if you think about the spectral space, you can see here there's a few different dyes. You can see there's something like Brilliant Violet 510, and you can see that broadly emits across the violet spectrum, but it's not really cross-excited, but it is using up some detectors that you might otherwise be able to use. And then there are other dyes like PE or PE tandems as well as PerCP that are cross-excited by both the blue and the yellow-green laser. And again, that takes up space that you could use to detect biological markers if you weren't using up both the blue and the yellow-green spots. And so in addition to that, when you're looking at building out a large panel, you can see here, this is showing you a single color with APC size 7, and in part A it's showing the uncompensated, and this is from the University of Chicago blog, looking at what is happening in terms of spread. So uncompensated, again, you see this where it looks like PE size 7 would be also expressed without the compensation, and then part B is showing ideally what would happen if you had compensated properly. So you can see that it looks like everything is expressed in APC size 7, and there's no change in the background for PE size 7. But in reality, what happens is that there is this compensation, spillover spreading error, and this isn't related really to the compensation. It's related to, basically this, the way that occurs. So in reality, you're still compensating, but you have this broader area. So if you had, for instance, something expressed in PE size 7, which was not expressed very highly, it would be hard to separate that biologically, whereas if it was bright, you'd be fine. So again, this affects your resolution. And in addition to all of that, all of these dyes, as we're building to more and more parameters, you think about how much hardware innovation has happened, especially really, in the recent space. So, early on, there were cytometers like the Fax Caliber, and then expanding to the Fax Vantage with 12 colors and LSAR2 with 18 colors. But now with the advent of, something like the Fax Symphony, which has a number of detectors off a conventional instrument, and then the Bigfoot, Aurora, and ID 7000, which are using spectral cytometry, this really is putting a lot of pressure, as you move to more and more parameters, when you're trying to detect similar fluorescent labels. You actually have to start dealing a lot more with non-ideal dye performance than if you can just position them away from each other in the spectrum. And to look more, in more detail at this idea of spectral cytometry, you can see this is just showing you two different dyes that have been acquired on the Cytek Aurora. So you can see at top there's APC and the other Alexa Fluor 647. And generally, when you're on a conventional instrument, you would just detect these in a similar channel, but with spectral cytometry, you can actually distinguish the two because you're basically taking advantage of non-ideal dye performance. So you can see that there are areas where these are, more different from each other when you get into, say, parts of the violet spectrum or other parts, as you look throughout that. But the truth is, a dye signature is still this result of non-ideal dye performance. And so while it will theoretically increase the number of colors in your panel, at some point the spread will also get contributed, and so you'll be limited at what you can actually use. And so one of the first things that we tried to do in looking at kind of a new system for dye design is to think about whether better performing dyes could expand the number of colors measured without increased spread. And so up top, you can see that there is, an imaginary dye called perfect blue. And why is it perfect blue? Well, it, is only detected in one of the 64 available detectors. It doesn't, it's not cross-excited, and, there's no sort of real decay. It's just, on or off. And that's not really how it works in terms of physics. So, what you have at the bottom is a tad less than perfect blue. And so the idea here is that, instead of breaking the laws of physics, you still have a very ideal dye in that you have a bright signal and then sort of a rapid decline, but not just this sort of on-off situation, which would be more of this, imaginary dye. And so, one of the things that I have tried to do is to really understand what's happening in spectral cytometry, and if you can design, cleaner fluors, what does that do in terms of this panel design? And for those of you that may or may not have been to some of the previous talks that we did under the umbrella of Phitonex, and now having been acquired by Thermo Fisher under the Thermo Fisher umbrella, you can see that this is taking originally a 40 color panel and then looking at where the issues of spread are, removing the dyes, and then building, adding in additional NovaFluor dyes with the antibodies conjugated to them, and then testing to see what happens, and what works and what doesn't work. And, so, we could get to these 45 colors, but the real question is, what is the deeper biology? And are there places where this is actually a problem in terms of the panel, and so the unmixing doesn't quite work the way, that we would like it to? And so really trying to get at this idea of the good, the bad, and the ugly. And you can see here, I have this image of a stagecoach to kind of think about spaghetti westerns. They are sort of done in these serialized ways. And so, the point of this 45 color panel is not just to say, okay, well, it's done and there's no more issues, but really to understand better what works and what doesn't. And I think what doesn't work is actually, really helpful for all of us to understand what is happening with spectral cytometry as opposed to conventional cytometry. And the problem with building to higher parameters is both the limit of the detectors available for the unmixing. So, even if you have, say, 64 detectors, you need some amount to be able to detect changes in the dyes. So, what are these signatures? So if you're using more and more of the detectors, that can be an issue. But also this idea of increasing spread. So any of sort of the problems in the background of what's happening with the dye, that can contribute overall to the resolution that you have biologically. And so the goal is to really understand how pushing towards a theoretical maximum number of parameters affects what you do with panel design and also the resulting biological resolution. And so stepping back from that, one of the things that we did, so this is showing you the Phyton platform, and what that is actually a new way to sort of think about developing dyes and then testing that out in these bigger flow panels. And so what you can see is that there is initially the single-stranded DNA, and there are donor and acceptor dyes that are on different strands. And then because the DNA has a sequence specificity and these dyes are conjugated, you can actually control the distance of the donor and the acceptor. And so this allows to really optically engineer and generate dyes that are engineered to have specific excitation and emission, or even increased brightness by design. And you can see here, for instance, the top one is showing you a 2-cluster version, and the bottom version is this 4-cluster version. And so what that means is that you can continue to build out in terms of brightness. And so then these can then be attached to antibodies, and you can look at these labels. And so in terms of thinking about the process of designing those labels, really had to step back and say, what would the requirements be if you could start from scratch with a dye? So you'd want it to be bright and clean, silent in the violet, and really limit laser cross-excitation. You'd want it to be consistent and stable, so if you're using it from one time to the other, that you don't run into any issues. And ideally, you'd like to be able to control brightness. And so this is just showing you the current dyes that are available. So this is showing you basically how we've kind of looked at targeting. So you can see that there are dyes that are excited by the blue, the yellow, green, and the red. We have not built since the UV and violet yet, but I would say, stay tuned for that. And you can see that in this case, what we've tried to do is to build along the spectrum, sort of looking at the aurora as a model in terms of, how many dyes can you build, and then also being able to separate out, for instance, what's detected off the blue and the yellow green, since that is an issue for PE and PE tandems. And so stepping back before going into the 45 color data, I want to talk a bit about how we've really targeted approaches to fluorescent label design. So what you can see here, for instance, is that, one of the things that I mentioned previously is that the blue and yellow-green excitation doesn't have to, what you have with PE and PE tandems, where they're excited by both of those lasers, doesn't have to be true if you actually engineer the excitation and emission. So that's one of the places we've tried to take a look at. Also, we're able to engineer the brightness. And then a few things that I think are not something I had spent as much time thinking about, really building to a longer Stokes shift. So understanding how, as you get more red shifted dyes, how those are more difficult to develop, but still very important in terms of being able to use the full spectrum. And understanding how the cytometer laser power and available detectors affect dye performance. And so just to start with the first one, this is showing you PE Dazzle and NovaFluor Blue 610. And this is not showing you the emission, but rather the normalized absorbance or excitation. So what you can see here is that, PE Dazzle is, a great dye in terms of being bright, but if you really think about, being able to use both the blue and the yellow-green lasers to excite that, you can see here that in the red that this would be excited off of all of those, whereas something like NovaFluor Blue 610, you can see is excited primarily off the 488 nanometer laser and not so much off of the yellow-green, whereas NovaFluor Yellow 610 is excited primarily by the yellow-green laser and not so much by the blue laser. And so what this does is allow you to really get this 2-for-1 idea. So instead of taking up 2 detectors with PE Dazzle, you can actually separate those out and actually measure 2 different markers instead of just 1. And there are people that say, well, I don't really have this issue, and that's because a lot of designers of instruments have designed around this problem rather than build dyes. So say on a Fortessa, there's often a trigon, and there might be a detector, say for FITC, PerCP, and then your scatter. But the reason that is because there are a number of PE and PE tandems that would be excited by both, and so you wouldn't be able to resolve that. But that doesn't mean your instrument couldn't do that. That's just existing dye performance. And so, I've talked about thinking about the separation of the blue and the yellow-green, but also remember it's important for these to be consistent and stable. And so what you can see here, these are showing 2 different lots of the NovaFluors. And what's really interesting is that, for instance, if you look here at the graph for NovaFluor Blue 530, you can see that it almost looks like there's just one line here. And these are 2 different lots, and that's in terms of how similar they are from time to time, from manufacturing batch to manufacturing batch. And so they really are consistent and stable. And this is in a lot of ways because instead of having, say with a PE tandem dye where you have to deal with where, say a Psi dye attaches, say if you're doing PE Psi 7, it might attach in different places. And so you have more of that spread. because you're attaching the donor and the acceptor at specific positions and you're able to control that, that really changes how these are consistent and stable in terms of that process of testing from lot to lot. In addition, if you look at the antibodies, you can see that the NovaFluor conjugated antibodies also really exhibit a lot-to-lot consistency. And so this is just showing you a titration. So you can see when you're in, the dose range and then reaching saturation in terms of the concentration, you can see that even though there's two different lots, the saturation and all of that is quite similar in terms of how they perform. So another thing that we've looked at because of the Phyton platform is that the NovaFluor dyes are stable at room temperature. And so, the way to test this, what we did is take antibodies and basically wrap them in aluminum foil and either leave them at room temperature or 4 degrees and look at how stable basically the dye is over that time period. And what you can see here, I like to think about this sometimes as the absent-minded graduate student experiment, because I certainly have dealt in my days with someone forgetting, leaving the antibody out. And even if you leave it out for these 14 days, you really don't see any differences between what's happening at room temperature or 4 degrees. And so not only are they consistent, but you can see that they're stable over this extended temperature incubation. And so in addition, there's this question of, okay, if you fix the cells, what happens? And this has been something that's been tested a lot with COVID. So, if you can't run your sample right away and you leave it in fixative, does that change what you see, or do you start getting more background? And you can see here that when you're looking at fresh cells versus fixed for 14 days, there's really no differences detected in those samples. So again, very encouraging in terms of how consistent and stable the dyes are. And then thinking about engineering brightness, so I mentioned before that there was a way where we can add more fluors onto, say, the existing structure. So our normal phytons, which are, compose the NovaFluors, are on these 2 clusters. So basically 2 of the 4 arms of this cruciform structure. And so another way that we can also build out brightness is actually, changing somewhat, the idea of what dyes are there and looking more specifically. So if you take this analogy of engineering for brightness, with a light bulb, you would have 100 watt, 200 watt, or 300 watts in terms of being able to change the same light. And so the same idea of being able to measure here. So we have thus far the NovaFluor Blue 610 at either a 30S or 70S version, and the NovaFluor Blue 660 at a 40S or 120S version. And what those are measuring is actually measures of the separation index. And so this is really a surrogate for thinking about brightness. And so something that is higher in terms of looking at, say, the 30S version, you can see here, and this is measured on an aurora using CD4 as the antigen. And you can see with the same antigen, you actually have 2 different brightnesses for the NovaFluor Blue 610 and the NovaFluor Blue 660. One thing I want to draw your attention to is that these are not exactly the same dyes. So rather than build with a 2-cluster versus 4-cluster, in this case, they're different component dyes. And so these, are very similar, but not entirely the same. So you can see the similarity index, which would give you that idea. So it's not 1, which would mean they're the same, but rather 0.97 and 0.96. So you could actually, add this dimensionality in terms of non-co-expressed markers and be able to look at those that way. Another great challenge that we've really been building towards is thinking about a long Stokes shift from the defined primary laser. And so when you look at this, you can see that there's the primary laser that's then exciting the donor dye, and then there's this high spectral overlap. And so it's very easy to see how you could have something that is being excited, say, from the blue laser, and then there's this donor, and you actually get emission further down the spectrum. But as you build out to more and more brightness, so you can see here at the bottom, you still have that donor, but now you don't have that overlap. So in terms of getting to a more red-shifted dye and getting this fluorescence, it's more of a problem than I think a lot of us spend time thinking about. And one of the examples I wanted to show here is just where you can see that when you look at, say, the yellow-green, there's this gap between YG5 and YG9. When you look at the red, this is R4, between R4 and R8. And again, those are just sort of jumps in terms of the detectors. And so building to these places where they're more red shifted is, a bit of a challenge. And so one of the things that we really wanted to try to attack with the system that we have. And so if you think about this, the goal in terms of this long Stokes shift from a defined primary laser is a bit like when you're throwing the football across the field in American football. So you can see here, if you're thinking about going that shorter distance, it doesn't take nearly the effort in terms of the throw. It doesn't take nearly the effort in terms of catching it. And that's really what we're doing, is taking this long distance across the spectrum and, trying to avoid any cross-excitation. And then ultimately what you're dealing with is that the acceptor of a lot of these Cy7 being an example, it's larger and tends to be more unstable, so susceptible to oxidation. So really building out dyes that are consistent and can, actually do this red shifting and allow you to use more of the spectrum is pretty important in terms of thinking about dye design. The other thing is that dye performance is really related to the instrument configuration. So a lot of work we've done has used the Cytec Aurora spectral cytometer as the readout. But what you can see here is that there are the 5 lasers if you have that instrument, and these can really range in terms of what is the excitation. So for instance, there's the 20 milliwatt UV laser, and then you go up to the 100 milliwatt violet laser. And I'm just showing you right down below that really when you look at a lot of different cytometers that even have lasers of these wavelengths, you can see that there's a lot of differences in terms of what you tend to see. And this is just taking a few cytometers, including say an Attune, a Fortessa, a few others, and really say, okay, what's happening here? And then also, I guess when you think about the Aurora, it's interesting in that the signature that you're going to see in terms of the dye performance still is going to be slightly different than another instrument, not only because of the laser power that you have, but also thinking about APDs, which is just similar in a way to detect the photons. So you can see that APDs are detect better in the red, and that's what the Aurora uses for the system, whereas PMTs detect better in the UV and the violet. And so when you're looking at designing for, say, 2 different instruments, and in this case, you can see that, if you think about, for instance, the Bigfoot spectral sorter, that it's a very different setup. And so designing for the same, designing to limit cross-excitation and also brightness can affect this process. So the Bigfoot has these 100 milliwatt lasers across the spectrum, and it does have, if you go to the 7 laser configuration, you have these additional lasers. And so really thinking about, if you're designing for dye performance, where is the cross-excitation, and does the brightness of some of these affect that process? So one of the things that we have, depending on which instrument you use, is something like the Nova Blue 660, the original 40S version. There are places where it actually can look brighter in the red. And again, that varies from instrument to instrument because of the fact that, there's, again, this better detection on the red off of the APDs on the Aurora specifically as compared to other instruments. And so the signature really is going to be related to the instrument, and thinking about the dye performance, that's, really important to, as we are designing new dyes, to really say, okay, does this work for this instrument, or even think about whether we should design something different for a different sort of instrument. And so taking a step back from all of that, thinking about what's happening with panel design. And so now I've talked a lot about different dyes and what they do. What you can see here is, a number of issues that you're contemplating, even if you're not, thinking about how that is all working together. So there's, the issue of spectral overlap. A lot of times we're dealing with that with compensation, and sometimes that is sufficient, and other times you can also have spillover spreading errors where you actually affect, how much you can actually detect. So, have you lost the resolution, and so these dimmer markers can't be detected. Matching up antigen density to say, if you have a highly expressed antigen versus a lower expressed antigen, matching that brightness. So for a lower expressed antigen, you want to make sure you have a more highly, a brighter dye, and vice versa. So these are all things that kind of go on in the background in terms of thinking about designing your optimal panel. And so as you're building to more and more colors, spillover is really the enemy. And if you think about the range of detection, there are issues with electronic noise, autofluorescence, but spread is the main thing that really affects what you can detect. And you can see here at the top, just to remind you of this idea that, for instance, in this case, even with a single color APC size 7, it's actually affecting what you can detect off of the PE size 7. And so it's acting as the perpetrator, and PE size 7 becomes the victim. And if you have something bright enough that you're detecting off the PE size 7, that may not be an issue, but if you're trying to detect something dimmer, that can be an issue. And you can see below, this is just showing 2 different NovaFluor dyes, and that there is not in adjacent channels on the attune, and you can see there's really not so much of the spillover spreading error that you're seeing in this particular case. And so, in previous talks, I have shown data where we built to 40 colors using the Aurora backbone panel. And so they had built to 35 colors, and in that case, this is before they published their 40 color work. And so one of the things early on that I wanted to test was, what is happening when you look at, say that 40 color panel, where are the issues? And then, if you try to increase that to 45 colors, how do you do that in a way to really think about what's happening? And so what you can see here so basically taking that, original 35 color panel, removing one of the labels that was not commercially available, so 34 colors, and then taking that to 40 colors, and then looking at, some of the places where there were issues. So in this case, BB515 and FITSI, there were some issues. Once you added NovaFluor Blue 530, they're already 0.98 similar, so that was a place of an issue. And then really looking at some of the other places. So as I look forward in terms of removing dyes to lower the spread, you can see here again, I mentioned that just by removing the Brilliant Blue 515, you can see that you've reduced a lot of the spread into the FITSI channel. And what this really is saying is, what is the sort of background that starts to creep up that affects what you can actually detect there. And so, when that's decreasing the change in the spectral spread, that means that you should be able to gain more resolution in that process. And so the other 2 places that I targeted was the BV510. And again, that, because it spreads more throughout the violet spectrum, it did help some, but more across the spectrum and not so much in one specific place. Another thing that I removed was the C-Fluor 568, and you can see again that there's this decrease in the spread. So you're able to actually detect a number of the fluors better off of the yellow-green that are excited by the yellow-green laser. And so to really identify then the next perpetrator dye to build to 45 colors, you can see here, in this case, PE Sci 5 is the next target that you would use, because you can see how this has, this is now not looking at so much the change in spread, but overall what's happening in terms of, the overall spread of the panel. And so you can see where that's more of an issue. And so having removed those 4 dyes, then the idea is that I wanted to test 10 additional NovaFluor labeled antibodies in a 45 plus color panel. And so just to step back in terms of how we did that process, what you can see here is that there are, different antibodies, and then these are the different NovaFluors there. And so if you wanted to see what the spectra was for any of the NovaFluors that I'm talking about, you can look at the Thermo Fisher spectral viewer, as well as Fluorofinder or FP base, and there's also the Cytec full spectrum viewer. So in terms of saying, okay, how similar are these dyes or not to each other? And so this is showing you one of the things that we tried to do to understand about designing into that panel, and showing the separation index for the NovaFluors. So again, this idea of measuring the brightness as well as thinking about the spread. And so something like Brilliant Blue 515 is, is a brighter dye and will give you more separation index, but it also has more spread. And so that will affect, what's happening in your overall panel. And the thing I would say is that really what you want to think about in terms of panel design is putting the fluor in that gives you the separation that you need, but you don't need the brightest thing if you're not taking advantage of that. So really matching up closely, okay, do I need something brighter here or something dimmer? And what is giving me the biological resolution that I want? And here, this is just showing you a few more of the NovaFluors. And you can see the ones that are excited by the yellow-green laser. So these are primarily excited by the 561 alone, as opposed to the PE and PE tandems, where they're excited by both the 488 and the 561, so starting to take up more of those detectors in your panel. And then one of the things that we try to do, and this is also on the Thermo Fisher website, is have these available so that you can really say, okay, if I've already got a panel, what does the separation index look like? And could I, for instance, be able to take advantage of this process and, switch to, say, a NovaFluor and open up another detector? Or do I really need that brightness because this is such a dimly expressed antigen? So understanding that process better. And then this last slide is just showing you what we have available at this point off of the red laser as well. And again, I mentioned that we're still building in the future to the violet and the ultraviolet. And so in terms of looking at what I showed you in the panel, we literally went through what we actually used for the custom conjugation process. And so here what you can see is that to do this, what we did was conjugate an oligo, a single-stranded oligo to the antibody, purify that antibody, and then leverage the one-to-one phyton to antibody labeling to determine the antibody concentration and the precise concentration of the NovaFluor dye required. And so with this customs process, what we do is actually remove any unlabeled antibody, and therefore you actually get this one-to-one labeling. And so you can attach the NovaFluor dye and not have any excess of that, around, or unlabeled antibody that will compete with the other parts. And so when you're looking at, the requirements for submitting this, generally we would prefer to have greater than 1 mg per mil of antibody to start with, and PBS with no azide, but these can be concentrated or buffer exchanged, so that's not really an issue. The main thing to be aware of in a custom conjugation is that any stabilizing proteins would be labeled in that process, and so that is actually could be an issue if there was a problem for background. And then, we can actually do these customs in-house, and we actually require a little more antibody. So if the customer is providing the antibody, this would be starting with at least 1 milligram of antibody. Or if you want to look at the final concentration in that amount, we recover about 40%. And the idea is because we're removing the unlabeled antibody, you need more antibody to start with, but it does give you a somewhat brighter signal for the same issue. So in terms of thinking about the currently available antibody conjugates, these are the ones that we have available. So, we have 5 in the mouse and 7 in the human, and these are available on all of the NovaFluor dyes. So we have 15 that were released up until now, and now as of in July, we're actually releasing the additional 4 colors. So every color can be found for every antibody, so you can really mix and match what you're using. And then last, just to bring this up before we go back into the panel, we also have conjugation kits. So if you wanted to say had 100 micrograms of unlabeled antibody and want to attach a NovaFluor, there's actually a fairly simple process to be able to do this. And so again, this follows a similar pattern to what we have in our customs program. So you can attach the single-stranded NovaFluor linker to the antibody. And then in this case, there's just these 2 pipetting steps after you buffer exchange then you can add the activation reagent and attach the single-stranded linker. The second step is to actually precipitate and remove any excess single-stranded linker and attach the NovaFluor dye. And then the third step is to actually mix this oligo-labeled antibody with the NovaFluor in the particular color that you want, and then you just mix them together and wait overnight, and they will anneal at 4 degrees overnight. And what this does is also allow you, for instance, if you bought more than one conjugation kit, you can actually leave it at this step after you've removed any excess linker and actually, match up 2 different colors if you wanted. If you had, 2 different NovaFluors, you can just make as much as you need at that time of the conjugate. And so that's really helpful in terms of, as I was even designing this panel and testing some things, there were some that weren't bright enough in terms of the separation I needed for that particular antibody, so I could mix and match pretty rapidly to think about that. And we are building to more antibodies being available, so I would say, stay tuned. That's one of the most exciting things about joining Thermo Fisher, is that we actually have that ability now. And so going back to the panel, you can see that in this case, what I did, again, was, take out the problematic dyes and antibody combinations, and then replace them with NovaFluors and add more into the panel to really detect, this broad immune panel. And then really looking in depth at, what's happening. So this is the spectral spread matrix, and you can see this is the column sum, so you can actually see where the places that actually have the most problems in terms of the detection. And one of the places the resolution was lost most, unsurprisingly somewhat, because there is, for instance, only there are 8 detectors off of the red laser. You can see that APC and NovaFluor Red 660 have a lot of spread, and probably AlexaFluor 647 contributes to this because you have these sort of 3 dyes that are in this very close proximity. And when I was originally designing this panel, we had not at that point, didn't have the similarity indices for these particular dyes. So I really wanted to see what happened. And what you can see is that, all of these are actually very similar when you look at their spectra. And in fact, NovaRed 660 is 0.98 similar to AlexaFluor 647. And so likely having these 3 together affects what you have in the process. And so then thinking ahead, so one of the things I would recommend is actually, saying, okay, maybe we don't include NovaFluor Red 660 here, and just use the APC and AlexaFluor 647 in this case. And then really looking at other targets. One of the most exciting things to see was how little spread there was across a lot of the blue laser excited dyes. And so one of the things I want to draw your attention to is that part of this happened actually in the way that when you're building out a 45 color panel, turned out we had ordered an antibody and it hadn't arrived in terms of being able to do the experiment. So I had to leave that out. But this ended up being fortunate because you really didn't have some of the issues that you might have otherwise. And so when you look at this, BB515 and NovaFluor Blue 510 are actually, 1.0 similar. When you look at their actual spectra, they're not exactly the same, but what this means is that they really can't be unmixed in a very good way. And even the FITC to BB515 is a pretty difficult unmixing, so 0.98 similar. But in terms of looking at these, NovaFluor Blue 510 and NovaFluor Blue 530 are actually the least similar, and so this reduces the spread and allows you to use more of the detectors across this range. In addition to looking at sort of what was happening across the blue laser, I wanted to look more closely at what's happening off the yellow-green. And so I mentioned before, you could see that the spread was quite increased with PE-Cy5. And that was likely because the PE-Cy5 is actually detected in 2 places that are very similar. So where NovaFluor Blue 660 and NovaFluor Yellow 660 are detected. So again, you could detect PE-Cy5 off of either of those, but since you're using both of those spaces, that can affect the spread. And so even though where you're seeing more of the spread is in Novaflor yellow 610, I would suspect that that's in part due to PE dazzle, where it would normally be detected off of the blue or the yellow green, but not necessarily both at the same time. And so that also is likely affecting that process. And so PE dazzle in this case might be the next thing to target to remove. And the last thing in terms of thinking about this is, is the Novaflor red 660 removal sufficient to remove spread off the red laser? And if not, it's probably worth looking at, removing one more dye from this region to really reduce the spread. And so what you can, as you're looking then at the panel, again, I already mentioned we've removed the dyes that affected the resolution off of the 40 color panel, built in this case to 45 colors. And then you can see that, one of the first recommendations would be to remove the CD141 Novaflor Red 660 to open up what's happening in the red. Then in this case, because I wasn't using the CD57 in FITC, you could actually move that off of Novaflor Blue 585 and separate that more, and then not create that bunching that happens with BB 515, FITC, and 530 and 510. And so last, removing the PE dazzle. As I mentioned, this should also decrease spread. You can move that to the Novaflor Blue 555. So this is what I would propose in terms of actually getting to a better panel with reduced spread. And so you can see that, here. So now you've moved CD24 over there. And so in this case, what I've done is taken CD44 from where it was on Novaflor Blue 585, CD141 away from the Novaflor Red 660, and then taken CD80, which was previously at Novaflor Blue 555, and taken those from the panel. And you still end up with a 43 color panel. And so again, it's really about thinking about what are you hoping to see and understand biologically. And so continuing to build this process. And so that's why I mentioned, this to be continued idea. So really thinking critically about what's happening. And so overall, stepping back from the specifics of this particular panel, if the goal is to really build a high parameter panel, the goal is really also to build a low spread immune panel without sacrificing the biological resolution. And so in terms of critical thinking around panel design, one of the things hopefully you've been able to see in this talk is that spectral cleanliness is more important than brightness for high parameter analysis. And so, as you get cleaner, spectrally cleaner dyes, you can actually use more of your detectors and really open up what you can see. As I've gone through, iterative panel design is critical to remove spread and work towards higher dimensional biological analysis. And so really thinking about, have I built the best possible panel, or are there places where I really need to look closer at spread and improve what I'm able to resolve biologically? So I would say, building any of these panels is a process of really thinking critically about matching, the antigen with, again, the brightness that you need. So not using more brightness than you need, which can increase spread in other areas of the panel. So in the spread is really, what's happening off of the non-primary detector. And so thinking about, continuing to improve that panel is really important. And then also you can really utilize the blue and the yellow-green detectors in a way that you haven't before in some of these cases, because you're building around the cross-excited dyes with the PE and PE tandems, instead of saying, okay, well, we can't use that detector and we have to use something else here, really thinking about getting this 2-for-1 idea off of one floor. And so in the end, it's not about how many colors that you're able to detect, but the resolution of the underlying biology that matters. And so I just want to emphasize that, because I think it's easy to get caught up in the idea of building some more colors and being able to detect all these things. But if you don't have the resolution, then you really don't have, good science, and that's what's really important, whether that's on a conventional cytometer or a spectral cytometer. And so just really thinking about, not just the specifics of this, but rather, how can we continue to build better panels where we're getting the biology that we're interested in understanding. And so with that, I'd like to thank everyone for attending the talk and open up the floor to any questions that anyone might have. Anson will be the moderator and can help, we can discuss any questions that you might have. So thanks so much for your attention.
Thank you, Seddon, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So we've already got a few questions, so we'll get started. The first question says, I use EDU Click-It kit, which has a fix, perm, and a catalyst-induced reaction. I see major shift to the right when I gate for CD4 and CD8. They appear compacted with this kit. How do the NovaFluors work after fixed perm?
Great question, and this is one of the places where we need to actually do the experiments to test it out. So I think in terms of the specifics of the kit, that just basically these two things need to be brought together and tested. But in terms of, I think labeling the excess cell, so if you're labeling extracellular and then doing a fix and perm, that, seems to work well with the NovaFluors. There is, I don't think you're doing it in this case, but if you were permeabilizing and doing intracellular, there are some additional blocking steps at that point, but that wouldn't be what you're talking about in terms of this particular kit. So great question. Just need to do more experiments on that.
Right. So I think we may need a little more clarification on this one. But how does this affect the diagnosis for patients, CD markers, HIV, dosing for Rh immunoglobulin?
Okay. Yeah. So I'll just try to speak in general to the concept. I mean, I think, one thing that spectral cytometry is really useful for is thinking about limited samples. I know that I actually used to do some HIV research, and one of the things that you're dealing with sometimes is if you have a question and you have longitudinal samples, it is really helpful to be able to use more markers to really look at what's happening. And so understanding that when you only have, say, one or two tubes left and it's very precious, I think that actually is a place where spectral cytometry is going to continue to play a big role, HIV, cancer, things like that. And then I guess in terms so feel free to throw another question if I'm not answering this. So I'm just kind of thinking about what, things might be there. I think, one of the things that's important in, being able to use your sample fully with spectral is important, but also making sure that the panel is really, well designed, and that you're not picking up something that, may be say an artifact if you have, say, spread or something like that into the channel. So I think sort of the two ways you really are able to use more of your sample, but you may have to take more time designing, say, these bigger panels. But once you do that, it should be good to use. And something I didn't get into during this talk, but also just, being able to look at that many parameters and setting up, okay, is this working? And I think one of the barriers going forward will be developing, say, master mixes or things like that. When you think about staining with 30 or 40 colors, that certainly is an issue, just to make sure that you have, when you're doing the spectral cytometry, you are having to do all your controls at that same time, or at least make sure the instrument is consistent. So.
Do you have any specific thoughts about some of the more recent PE and APC conjugates and how they interact with NovaFluors? For example, PE FARE 600 or APC FARE 810?
Yeah. Well, in both of those, what you can see is that they're focusing on a similar type of problem, which is getting to this red-shifted area where there just aren't a lot of dyes in that area. So obviously a really important thing to address, in terms of getting into this space where you aren't actually using up some of the spectrum and getting there. So, I think in terms of PE Fire 600, you do have, again, this issue with the blue, yellow, green split. So in terms of the novaflores, I'm going to have to think about what wavelength that is at. But if there are novaflores in there, you can actually, be able to use that 2-for-1 issue, whether that's spectral or otherwise. And so the other thing, I think these are, good in terms of reaching that further part of the spectrum. They are bright, so sometimes they will have some off-target, cross-excitation or otherwise, but I don't think there's really any issues in terms of using them in a panel. So as long as you're getting the resolution that you need and not introducing spread, you should be able to use those in a panel together. So hopefully that addresses your question.
Yeah, great questions. Please keep them coming. Do DNA-based dyes have an effect on DNA binding reagents?
Yes, they do. Really good question. So we recommend using fixable viability dyes with novaflores. And Anson and I early on actually spent some time troubleshooting when someone had some issues staining, because unfortunately a dye like DAPI, 7-AAD, perfidium iodide, they will bind to the DNA structures because they are DNA intercalating dyes. And so if you can use either some of the calcium based viability dyes or the live dead, some of those, basically those will bind and to the cell and not be the DNA intercalating, but you really can't use those together. And if you did use those, what happens is it either quenches or alters the fluorescence. So we spent some time troubleshooting that. So that is definitely an issue that you need to be aware of.
Here's a really good question about Purple Rain. When are you planning to introduce 405 nanometer probes?
I will say that it's definitely a work in progress and something that is pretty exciting for me, because one of the things I get to do, and Anson, also, does some work looking at these, but basically we do a lot of the dye vetting. And so part of the process is, testing, does this dye either brighter or have less spread than things that are existing. So I will tell you that those experiments are in progress, and there are some interesting candidates, but just stay tuned. We're definitely working there.
Are there any ways of combining cell cycle analysis or measurement of DNA content with staining with novaflores?
Such a good question, and something that I certainly have spent some time thinking about. And I know that Sean Burrows, who has also worked on the dye design side, one of the things we've tried to think about. So I think it is an open question. I don't have an answer now, but I'm certainly it's one of the things that we're really interested in, is whether there can be a way to kind of balance it out. Currently, there's not a great way to sort of use an intercalating dye that you would be using in cell cycle analysis. You could use things like, say, Ki-67, but I think in terms of probably what you're really asking, I think that still is part of the next frontier. There are, I think, some other cell cycle dyes, and that's one of the things I really want to kind of look at, is see if there are other dyes that can be used with the novaflores there. But in terms of intercalating dyes, unless we find a way to kind of shield the structure, that's just going to continue to be an issue. But great question.
All right, so we've got more time, so if anybody has any more questions, please continue to drop those into the Q&A box. Can you tell me what the size of the phyton is?
Yeah, so it's 20 nanometers across, so it's not exactly flat, but in terms of that sort of curved surface, if you think about the cruciform structure, and it's about 150 kilodaltons. So it's actually somewhat smaller than PE, which is, I think, around 220 or 250 kilodaltons. But yeah.
So can you tell me a little bit more about the structure of the novaflores? Are they tandem dyes?
Right. It's a really good question. So when you think about, the NovaFlora structure, what it is, it gives you really this ability to position very precisely your donor and acceptor dyes. And because they're actually conjugated at a specific point of the DNA, they are precisely placed. So on one level, you can think about, a tandem as being a dye where these are placed in certain positions to allow FRET to happen with a donor and acceptor, and so that's true. But the most tandem dyes you have to conjugate this directly and can't really control where the donor and acceptor are, or sometimes how many there are. Say if you think about PE-Cy7 or something like that, it may attach at different places and have different numbers of those. So you really don't get that consistency and stability from time to time using them. And so we sort of think about this as really just a different type of structure, because what you find with novaflores, because they are at these specific points in terms of the donor and acceptor and where they're positioned, they're actually very repeatable, and they seem to be a lot more stable than a lot of the existing tandems that are out there. So I would say it's a different class of dye.
One of the things that was mentioned was cell blocks. Is that an FC block?
Really, yeah, good question. So cell blocks is not an FC block, and it's also not the same as a super bright or any of the brilliant binding buffers, which prevent interactions between dyes. What it does is actually prevent non-specific binding of dyes to the cells. So cells will actually can sometimes bind to the dyes, and so that's what it actually does, is interfere with that process.
All right, looks like we have time for one more question. As I understand correctly, you are looking into spreading the novaflores into the violet spectrum. Do you think that scientists get more out of novaflores instead of super brights or brilliants?
Yeah, it's a good question. I mean, I think the point is that hopefully as we do expand that, what we're trying to look at is what dyes are currently out there. So trying to say, okay, does it have a cleaner spectrum, or are we getting to something that's brighter and more specific? Sort of trying to look at both of those before releasing them. So one of the great things about the platform is we can test a number of things. And so what I would say is, by the time we are, looking at that, we are also comparing that to the super brights or the brilliants. The other thing is that in some of these cases, if you are dealing with a really bright dye and don't have, much separation, say with a transcription factor, I think, ROR-gamma is a good, example, where you just have a very small shift, even when you have cells that are specific. And so sometimes you need that brightness. And so I would say sort of it's balancing what you need in your spectra, in your panel, in terms of, do you really need, separation of something that has very little antigen density, or, do you really want that cleanness because you're building out the brighter, this sort of bigger panel. And so we do try to compare it and say, okay, is this particular novaflora, how does this compare to what's out there at that part of the spectrum?
All right, I'll sneak one last question in because I know the answer is going to be very short. Have you tested some of your 40 plus novaflora panels on the Sony ID 7000?
Not yet. I would love to get access to an instrument. So that's, it's always exciting to try to test one of those things, but currently I don't have access to a Sony ID 7000. So hopefully at some point that would be something we could test.
Thank you, Seddon. Do you have any final comments for our audience?
I just want to say thanks so much for coming, and these are fantastic questions. So really appreciate the engagement and conversation back and forth with all the questions. So thanks so much for attending.
Thank you again, Seddon, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.